Okay. So my name's Helen Prior. I'm from the National Centre for the Replacement, Refinement and Reduction of Animals in Research. So that's the NC3Rs in London. So we are the UK Centre for the 3Rs. I was going to introduce my co-chair, Andrea, as our first speaker, but unfortunately she had a problem with her plane last night and she wasn't able to make the meeting. So I will make an attempt, a good attempt, hopefully, at giving her slides in the next talk. So this symposium is designed and supported by the board of the Safety Pharmacology Society, where I had a term as secretary and then another as a normal board member around the time that this symposium session were being considered. So we'll explain more about the discipline of safety pharmacology in the next talk, but we first of all wanted to provide insight into some of the new technologies and approaches that have evolved to improve both risk, which is safety assessment, and predictivity for translation to the clinic. So the Safety Pharmacology Society was inaugurated in the year 2000, which was the same year that our main regulatory guideline, ICH S7A, was published. So, of course, in the last 20 years, a lot has changed, including more regulatory guidelines, and actually this has been updated recently, which we'll mention more in the next talk. But both cardiovascular and CNS adverse effects are still major reasons for drug attrition and withdrawals from the market. There are many drivers for change, including updated practices and approaches, such as availability of new and evolving technologies, some of which you'll hear about today, a wish to streamline packages and reduce animal use, and the need for non-clinical data that better translates to clinical outcomes. This is our current board of directors. Our president this year is Marie-Luce Rossils from UCB in Belgium. And you can see that Andrea, who should have been speaking next, is our vice president-elect. So she'll be president in 2025. Our society has about 480 members from around the world, representing 22 countries with 61% from North America, 34% from Europe, and 5% from Asia Pacific. As a society, our vision is to lead the global safety pharmacology community in the development and safe use of biologically active molecular entities by bridging across disciplines to predict, identify, and mitigate potentially undesirable pharmacodynamic activities in non-clinical studies and guiding their translation into clinical trials and beyond to benefit all patients. We achieve this vision through three main strategic goals: advancing the science, education, and partnerships. These are some of the activities of the Society. You can see we're going to have our annual meeting in September this year, and it's in Brussels, and this alternates between Europe and North America each year. We also provide learning opportunities via webinars, access to the Journal of Pharmacological and Toxicological Methods, and our accreditation program to become a diplomate in safety pharmacology. Both myself and Andrea and Henk as well, are accredited diplomats in safety pharmacology. The other major activity is to be the guiding voice for advancing the science in the field, to ensure we're providing the data that's needed now and into the future. This includes best practice publications, which in the past few years have focused support for the industry to interpret and implement changes associated with the new Q&As, which we will mention in the next talk. And with that, we'll get into the presentations for the workshop, and this is the agenda. And we'll have the time at the end of each presentation for audience questions for each of the speakers. And I'll invite you to, you can either input questions via the app, or you can come up to the microphones in the middle of the room. So I was going to introduce Andrea, the first speaker at this point, but obviously she wasn't able to come. So as I said, my name is Helen Prior. I'll introduce myself. I'm not sure I've done this before. I'm from the NC3Rs in the UK. I've worked there for about 7 years now. I'm a program manager for drug development. And more recently, I'm now a scientific advisor part-time. So sorry, I've lost my train of thought. Before I was at NC3Rs, I was working at AstraZeneca and Covance, which is now LabCorp in the UK. So I've been in the pharmaceutical industry for all of my career. Okay, so I'm going to give Andrea's presentation now, which is the keynote presentation entitled Advances in Safety Pharmacology for Improved Risk Assessment and Translation of Cardiovascular and CNS Adverse Events. So first of all, what is safety pharmacology? So there's three aspects of pharmacology. The first is primary pharmacology, which is the sort of efficacy, the desired mode of action and effects of a substance in relation to its desired therapeutic target. Secondary pharmacology, which is around mode of actions and effects of a substance not in relation to its desired therapeutic target. So does it interact with other receptors, other enzymes, other transporters, etc. And then we have safety pharmacology, which focuses on the potential undesirable pharmacodynamic effects of a substance on physiological functions in relation to exposure in the therapeutic range and above. Okay, it's not progressing. Slides on. Okay, thank you. So this is a paper that was published in 2014, so it's all coming up to 10 years old now. And it looked at various drugs in the AstraZeneca portfolio and looked to see the reasons for attrition and which of these were safety and which were from efficacy. And actually this figure shows the different target organ systems which were involved in safety failures, both preclinically, which are the red bars, and clinically, which is the brownie bars. And you can see that cardiovascular and CNS are major reasons of attrition at least 10 years ago. And actually, it's still the case today. So we have two major regulatory guidelines for safety pharmacology. That's ICH S7A and ICH S7B. ICH S7A was inaugurated in the year 2000, and this describes the general principles and recommendations for safety pharmacology studies to protect the initial clinical trial participants and patients. And it defines a series of studies, and it's specifically a core battery of studies. So this is studies which need to be performed on the CNS, the cardiovascular, and the respiratory systems. These systems are deemed as intrinsic to life. So were there to be a one-off large effect on either of these three systems, then that could be a massive adverse effect and definite for life. The next guideline was introduced in the year 2005, and this is specifically for cardiovascular assessments. And it defines a non-clinical strategy for assessing the potential of a test substance to delay ventricular repolarization. So it's looking at some in vitro studies with the HERG, inward rectifier potassium channel, in vivo cardiovascular study, and potential follow-ups. And what's important is for small molecules only. So central nervous system safety pharmacology is assessing drug effects on the central and peripheral nervous system. It's looking at things like behavioral changes, sensory motor reflex responses, motor activity, coordination, and body temperature. And we generally use tests called the modified Irwin test or a functional observational battery or other appropriate tests. And I'll describe these more now. So the Irwin test is named after the gentleman who introduced it in the late 60s. And it's most commonly assessed in rats. So the principle, it's a detailed scoring of behavior across multiple parameters and multiple time points following a single administration. So the first few observations are made the animals in the home cage, so they're undisturbed. So we're looking at things like, do they look normal? Is their fur ruffled? Is there normal urination, defecation, and things like that? The animals then moved into an arena and looked at motor parameters, so locomotor behavior, so is there ataxia? Is it moving, not as you would normally expect? Posture, is it rearing a lot or not, etc. And then we go into the manipulation. So we're looking at reflexes such as grip strength, sensory systems, and body temperature. Other models which are often used to measure CNS is the spontaneous locomotor activity and rotor rod models. So the spontaneous locomotor activity is generally performed in rats. So the picture at the top left is a rat is put into this arena, and there's beams across the bottom and along vertically and horizontally, so you can measure the movement of the animal via the breaking of the beams, and if they rear up, it breaks the beams above them as well. And so it measures, you put them into this new novel arena for about between 30 minutes and an hour, and you look to see if there's any difference in activity. So the graph on the right-hand side is for olanzapine, which is an antipsychotic. The black line is for control animals, and it's normal for there to be a high level of activity when you put the animal into the novel arena, because it's going to be exploring the novel arena. And then this dips off over time, as you can see, after about 20 minutes. But with this drug, you can see there's a dose response decrease in activity of the rats with this drug. The rotor rod is similar to a human treadmill. So the animals, so the rats, and you can get smaller ones for mice as well. So the bar in the middle is a rotating cylinder, and the animals obviously have to walk to keep their balance as the cylinder moves around. And so it's an assessment of motor coordination. And you tend to put them on for about 2 minutes. And as they fall off, it's only a short distance, they break the time pedal below the cylinder. So this is the same compound, and you can see that with high doses of this compound, I think it is, it's the same compound, that there is some falling off and some reduction in the motor coordination of these rats. A more novel and newer technique is an automated behavioural monitoring, which can be done in the home cage of rats and mice as well. So automated, non-invasive, the animals are being monitored for 24 hours. Basically, there's cameras focused on the cage. Each of the animals, you can have three rats or mice in the cage at the same time, so they're in their home environment, they're group housed, and they're not being disturbed. Each animal has one of these tiny little RFID chips implanted, and that measures their activity. You can follow them along the grids, the black and white grids, and it also measures their body temperature. So there's infrared lighting, so it can be monitored overnight as well. And this system was developed actually following grants and funding from the NC3Rs quite a while ago, probably, when was it? Probably about 2013 that this project started, and it's in its final validation stages now. So the company that produces it is a Scottish company, actually, that was a spin out from Edinburgh University called Actual Analytics. And the idea of this project is that you can, it does actually apply all 3 Rs. So you can, it's a reduction because you can get more data from fewer animals. It's a refinement because there's less handling and disturbance of the animals. And it could potentially be a replacement because it could be, the data could be potential for simulation to replace actual control animals in some studies. Moving on to respiratory safety pharmacology, this is commonly assessed in rats, typically by a method called plethysmography. And these two chambers that you can see, the bigger one is for a single rat, and the smaller one is for a single mouse. And what you do is you put the animal into this chamber, and it has air flowing in and out of it, and the pressure within the chamber, it's a sealed chamber, the pressure within the chamber, it can be monitored. So as the animal breathes in and out, you get a waveform of the pressure differences within the chamber. And this can give you parameters such as tidal volume, which is the capacity of the lungs, respiratory rate, so the rate of breathing, etc. You can get non-invasive systems, so jacketed systems. So you can see a picture here of a dog wearing a plethysmography jacket. So this jacket has got two bands, one across this area and then another on the abdomen of the animal. And the difference in the waveforms is measured by inductive plethysmography, really. So you can include those types of measurements on other studies. It's non-invasive, so the animal can be wearing it for 24 hours, etc. And the other picture is a pig as well. So you can get systems for rodents and all the different non-rodents as well. So on to cardiovascular safety pharmacology. And the reason for the discipline really being being here today is about this arrhythmia called torsades de pointe, which I'll go into in some more detail here. The history of this arrhythmia, was first reported in 1963, and it's deemed as a kind of twisting of the points of the T wave, that was coined in 1966. And over the years, various drugs have been found to cause these arrhythmias. And the first drug was withdrawn in 1988, and a famous drug was tafenidine, like an antihistamine, which was withdrawn in the late 90s. And you can imagine drug development is a balance between risk and benefit. And if you have an allergy, a hay fever allergy, you're not going to accept a side effect such as arrhythmia. So the industry and the regulators have really come up with a range of different studies and requirements for QT interval and the potential for these arrhythmias to be thoroughly monitored before the first clinical trials. So underlying this arrhythmia, it was discovered the Hurg channel, which is an inward rectifier potassium channel, it underlies the potential for QT prolongation. So the Hurg channel is responsible for repolarization, so that's phase 3 of the action potential that you can see here. Okay. And lots of drugs interact with the Hurg channel and block of the Hurg channel, which can cause prolongation of the QT interval. So measurement and assessment of Hurg block is performed very early in drug development. Screening of hundreds, thousands of compounds through high throughput machines. And then once we get to the regulatory packages, there's a requirement within S7B for a patch clamp assay, which is considered to be the gold standard. So a manual patch clamp looking at the currents in the Hurg channel, Hurg potassium channel. Then the next study which is required is an animal study looking at the overall ECG. So as I said, the top ECG you can see there is a normal ECG. So it's got a QT interval of around 0.44 of a second. But when you have Hurg block, which is the longer QT below it, you can see that the QT interval is extended and it's now 0.63 of a second. Torsades is a rare event. There's not enough animals or patients in studies to predict it, but the QT interval is used as a surrogate marker for potential for torsades in people. So the study that we do to investigate this is one in non-rodents. So you usually do it in the same species that the toxicology package is being performed in, so mostly dogs, but you can use mini pigs or monkeys. It's a telemetry study, so the animals are non-restrained and they're instrumented to measure ECG and blood pressure, heart rate, and also core body temperature. So the animals are conscious, they're in their home pens, they can be dosed and then they're monitored for 24 hours or longer as necessary. And they serve as their own control, so they generally have vehicle and 3 dose levels in a Latin square design. Animals are very often reused. Most companies or CROs have a colony of instrumented animals, so you will run a drug through these animals, and then they have a washout period for around a month, and then you can reuse them. And the important thing to note is these standalone studies aren't needed for biologics or oncology drugs. So for any compound which is following ICH-S6 or ICH-S9, then we don't have to do this study. Here's an example of some telemetry data. This is from thyroidazine, another antipsychotic, and on the left is in the mini pig actually. So you can see that there's 20 hours of data, and we're measuring the QT interval corrected for heart rate. The black line is the control, and you can see that there's an increase in QT interval with this compound. The right-hand side shows a different compound. This time it's in dogs, and it's showing the blood pressure data over 6 hours. And what's of interest here is this spike that you see when people have gone into the room to take a blood sample. Obviously, it stimulates the animals, and their blood pressure and the heart rate goes up anyway. And so you have to probably ignore sort of 30 minutes of data until it comes back to normal. So this is we try and reduce the amount of blood sampling in a study. Because of these disturbances, and we want the primary endpoint is the cardiovascular parameters. So bringing the in vitro work and the in vivo work together, the left-hand panel here is the normal HERG, action potential, and ECG, and then the right-hand side is the prolonged HERG, action potential, and ECG. And as I said, many compounds block HERG, but actually very few of them cause torsade. So safety pharmacology studies are generally performed, so a lot of early screening for ion channels, etc., and you're going to hear more about that from Kim and from Max for in silico modeling in the next few talks. And then when we get to the regulatory package, so the entry into human package, then we're required to do the HERG in vitro study, and also most companies will perform an assessment of the calcium and the sodium currents as well. In silico modeling has been increasing over the last decade, and the other animal studies which are required are, as I've said, the non-rodent telemetry, the Irwin screen, and the respiratory study in rats. And very often, ECGs will be included in toxicology studies as well. And then when we get to phase 3, we're into the human clinical trials. And there's a possibility that there's a thorough QT clinical study which is required, and we're going to talk more as we go on about the potential to waive these studies now. But first of all, there's a difference between in vivo safety pharmacology studies and toxicology studies. They're similar but different. So the aim of the safety pharmacology study is to assess the undesirable effects of a substance on the physiological functions of specific organ systems, whereas the main point of the toxicology studies is looking at the histopathology following chronic exposure. So safety pharmacology is looking initially at CNS, cardiovascular and respiratory, but there are models and tests for the other organ systems as well, sort of sometimes renal and GI assessments. Whereas a general toxicology study is an in-life assessment of clinical science, clinical chemistry, body weight, food consumption, etc. Safety pharmacology is typically single dose. General tox is repeat dose. And safety pharmacology uses an appropriate non-rodent species, often a single sex, whereas the general tox is looking at rodent and non-rodent species and both sexes. So the translational impact of safety pharmacology studies, looking at CNS, there was a collaboration of 4 or 5 different pharmaceutical companies, which was published in 2016. And they looked at the preclinical effects, which are identified in the CNS model, so in the Irwin or the FOB, and they compared them with the adverse effects in the first in-human clinical trials. And actually, the 5 most common adverse effects in clinical trials are things like pain, fatigue, dizziness, nausea, and headache. And they're not very easy to monitor in animals. An animal can't tell you that it feels nauseous or it's got a headache. There might be some indications of nausea if it doesn't eat, and dizziness if its gait is a bit irregular. But overall, there's not very good concordance between the preclinical and the clinical. The more serious effects, like convulsions and ataxia, are readily detected in animals. Cardiovascular translation, again, there's been a couple of publications, collaborations across the industry. So this one on the left was published in 2014, showing good concordance between telemetry dog data and human QTC effects. But less so for heart rate and other hemodynamic effects. The one on the right was published in 2019. I think this was just Pfizer data. And they're showing that dog and monkey data combined have good predictivity for the human hemodynamic effects. I'm a bit unclear about this data because it's not often that you would have data in both of those large species. Normally you would just be doing your studies in one of those species. So this is where I fall down with what Andrew was going to say here on this slide. So high-quality ECGs can also be included in toxicity studies via this system called jacketed external telemetry. So it's the animal wears a jacket and the ECG leads are non-invasive, just on the surface of the animal. You can also have other implanted systems for more accurate measurement of blood pressure. But that includes surgery, and also the animals require training for acceptance of the jackets. So novel in vitro assays beyond HERG. So there's microelectrode arrays using human cardiac cells. So they're either heart slices or cardiac cells, or cultured iPSCs. Yep, and there's also hybrid MEA impedance technology as well, which can measure functions, so more like the contractility rather than the sort of ion channel activity. And 10 years ago now, the health authorities initiated this SIPA initiative, which stands for Comprehensive In Vitro Pro-Arrhythmia Assay. And there's 4 components which people were asked to investigate and provide data for. And it's all looking on the in vitro side. So it's asking for measurements of all 7 cardiac ion, the main 7 cardiac ion channels. That data is used in silico to reconstruct the human action potential, and also looking at human stem cell derived myocytes, and then looking at data which is provided from the clinic as well. Because there are situations where even if a compound does prolong QT, if it also has multiple ion channel blocks, so if it blocks calcium and sodium as well, then these can counteract the hERG block, and then that compound is safe. An example of that is verapamil. So what has been happening in the industry is there's thousands of compounds have been screened for hERG, and if they block hERG, they've been stopped from development. However, had those compounds had multiple ion channel block, then it could have been that those would have been safe compounds. So we're moving more towards an overall picture and away from just hERG on its own. And this is where this new ICH Q&A has been developed. This was issued last year in February. And it's the first time that the S7B and the E14, which is the clinical regulatory papers, it's the first time they've been combined. So is QT prolongation still an issue in drug development? So from the FDA did a review of their study reports for the 5 years up to 2020. And actually, 81% of the time, there's no QT prolongation. So the industry has been doing really well to reduce this liability. But 19% of the time, there was QT prolongation. A lot of the time, this was for oncology drugs or drugs targeting the central nervous system. And you can see the other therapy areas there, which commonly have compounds which increase the QT interval. So there are 2 Q&As for the E14 available where you can substitute the thorough QT studies. So there are times when you don't need to do the study, and obviously this saves time and money for pharmaceutical companies. And these are under these 2 areas of the guidance. So there's use of concentration response modeling of QTc data, and if the clinical studies characterize a QT at greater than 2-fold the high exposure, then no positive control is needed. Or there's alternative study designs when there's no conventional QT assessment, for example, with oncology compounds. Yeah. I'm not sure what Andrew was going to say about this, so I'll just skip that one. So the new Q&A has introduced this concept of a double negative non-clinical data. So the non-clinical data is now super important because if you have a negative hERG study and a negative large animal telemetry study, then that data can be used to support a waiver for your thorough QT clinical study, which is obviously of great attraction for the pharmaceutical industry. And a couple of papers have literally been published in the last couple of months as collaborations between Safety Pharmacology Society and all the different pharmaceutical companies that you can see here. So one of the options is waiving the positive control in the clinical QT study. So the positive control isn't necessary if the following conditions are met. So either the data characterizing the QT response is 2 times multiple the high clinical exposure, or the new one, only 1 times the clinical exposure were covered in clinical ECG, but the non-clinical integration was double negative. Another new one is a double negative in the non-clinical assessment. So you've got a negative hERG model and a negative no QT prolongation in the in vivo study. What's important here is it needs to be of sufficient power to detect a QTc prolongation effect of a magnitude similar to the dedicated clinical QT study. So they're saying that is you need to be able to detect a 10 millisecond change in QT in your animals to be of similar sensitivity as the measurements in humans. And if you're below that threshold, then that gives you, some data to ask for a waiver. So the industry has come together and has been working on its best practice requirements for both in vitro and in vivo. These different requirements are explained very well in ICH training materials and in the 2 publications which were shown on the previous slides. But if there is HERG block and QT prolongation present, as I said, not all of this leads to arrhythmia. So follow-up studies to assess the torsade risk could be measuring other ion channels to see if there's multiple ion channel block, in silico modeling, and also cardiomyocytes. And if further information for this can be found in the training material from the ICH and these 2 publications from the safety pharmacology industry. So in summary, safety pharmacology as a discipline is to assess functional endpoints to allow safe entry into human. The core battery focuses on the CNS, cardiovascular and respiratory endpoints, and further refinements in CNS testing are needed to enable increased predictivity. But advances in cardiovascular in vitro and in silico tests will further predict arrhythmogenic liability for the cardiovascular area. And recent Q&As within our regulatory guidance do now enable the use of non-clinical studies to support thorough QT waivers. So these were the people that helped Andrea to produce this presentation. I hope I've done a reasonable job at going through her slides. So if there's any questions, I'll take them now. Thank you.
Thanks for that. A major problem in human pharmacology, measuring QT interval, is that it varies, as you said, with heart rate. And ideally, in any individual, you would generate different heart rates and a correction factor for that individual, which is not in practice possible. So one relies on population data, and you never know whether the QT interval is actually properly corrected for any heart rate in an individual patient. Which is, I guess, most of the time it's okay, but there will be occasions when you're not right, and we're always concerned about that. Now, I would expect that in animals that wouldn't be so much of a problem, but I wonder if it is.
So we do correct for heart rate changes in the animal studies as well. There's quite a range of heart rates over the course of the 24 hours, as you saw from the blood pressure. There's a spiking activity when people are going into the rooms, and at night, obviously, they're asleep and lower heart rates. So yes, there is. What happens is they generate an individual regression for each animal, right. And then so each animal acts as its own control, really, for its QTRR regression curve. And then that's what's used in the data analysis. So yeah, it's a similar process as what happens in humans.
So is there less variability from animal to animal than there is from human to human?
What would you say, Henk? Less variability in dogs than humans?
Well, for humans, because I'm more non-clinical, but there is...
It's quite a lot of variability, isn't
it? Yeah. There is some variability, but it's not that big.
Okay.
I think when you use the right correction, of course.
And
the is the best. That's what we use most of the time now in our models.
Because of course you don't have the polymorphisms in the laboratory animals because they're inbred and they're more controlled than the human population as well. Any other questions? Yeah, there's another one here.
I'm Gerard Ronger, clinical pharmacologist from the Netherlands. You are focusing on arrhythmias as an important cardiovascular safety problem, but I saw in your slide that with the attrition rate, that despite the preclinical research you're doing, there's still more withdrawn during the clinical development of drugs than during the preclinical development with respect to cardiovascular safety. So are we looking at the wrong outcome? Is there other cardiovascular toxicity that is causing this withdrawal? Can you comment on that, please?
Yes, so obviously we've got the other side, so we've got the hemodynamics, you've got your blood pressure effects. There's also the cardio pathologies as well. So so there's other cardiac reasons which are all lumped into that same column essentially.
And can you quantify that a little? So, because if that is really a large proportion of the problems, then we should not focus that much on QT, I think. Then
Well, that's why we've been moving away from it, because essentially, you know, not that we've solved the problem, but at least we can detect it. So, yeah, I mean, the toxicology community are looking more at the pathophysiology side, but yeah, I'm afraid that's a little bit outside of how I can answer the question. Okay. Sorry.
Thank you.
Okay, looks like we don't have any more questions, so we'll move on to our next speaker. Let me introduce our next speaker. The app has gone down and all my biographies have gone. Let me bring it up again. Come on. Okay. It's quite slow. Okay, so that's not working. Anyway, so luckily I know Kim. So Kim is a senior scientist at ApconiX, which is a CRO and research lab in just outside Manchester in the UK. And she has been, she did a postdoc at Durham PhD at Durham University. And she is currently a research senior scientist at ApconiX and is looking at different in vitro methods for seizure liability screening. Thank you.
Thanks for the introduction, Helen, and also thanks to the SBS for inviting me to speak to you all today. So as Helen said, I'm a senior scientist at ApconiX. We're a non-clinical toxicology CRO, and I've been working there for quite a few years now, and I've been developing in vitro methods for early detection of seizure liability. So first of all, so we're all on the same page, I'd like to define what a seizure is. So at the simplest level, seizures are sudden uncontrolled electrical disturbances in the brain. And normal activity in the brain relies on a balance between excitatory and inhibitory activity. So if this activity is disrupted, you get either too much or too little activity, and then we can have seizures. So within my talk, first I'll cover some background. So first of all, I'll define the problem of seizure liability in drug discovery. So why do we care about it? I'll go through the methods that are currently used to identify seizure risk and some advantages and disadvantages. And then I'll briefly mention the preclinical cardiovascular screening strategy, so HERG, etc., that Helen was talking about, and see if there's anything here that we can learn that we can apply for seizure detection. For the rest of my talk, I will discuss what I've been doing at ApconiX. So I've developed two methods for seizure detection. The first one is an ion channel panel for early detection of seizure, and then the second one is an IPS microelectrode array assay, or MEA assay, for seizure detection. I'll then finish by proposing an early discovery screening strategy that incorporates both of our assays, so how they can help within drug discovery. So to start off with, let's look at the problem of seizure and attrition in drug discovery, so some facts and figures. So issues with drug safety and toxicity are a leading cause of attrition in drug discovery. So here I've just pulled some figures from Cook et al., who reviewed AstraZeneca's pipeline. And you can see here in this graph that was in the preclinical and phase 1 phases of drug development, safety is the main cause of project closures. And after the cardiovascular system, adverse events in the CNS are the second most common source of attrition. So here I've got the same graph that Helen showed, showing the organ systems involved in safety failures. And you can see that for the CNS, most failures are actually in the clinical phase of development, which is where the stakes are higher in terms of money invested and also patient impact. So all of this points towards the fact that we need more predictive preclinical studies for CNS-related toxicities. Now I've got a couple more graphs to show you. The first one is by Orthier et al., who did an industry survey on the nervous system. So you can see on the graph on the left, it shows all the drug-induced preclinical CNS issues encountered by respondents in 5 years, and seizures right up there at the top of the list. And on the right-hand side, the graph shows the incidence of neurotoxicities in the FDA label database, where seizure also makes an appearance. And if we look at the big range of CNS toxicities that are on the slide, it's everything from gait abnormalities, salivation, visual impairment, suicidal ideation, abuse liability, etc., which will all be very, very difficult to model in vitro, whereas because seizure is related to changes in electrical activity, it's something that's quite tractable, so it's amenable to early screening, and obviously a problem that it would be nice for us to solve. So here I've got the current methods that are used for seizure detection. So starting from the left, clinical observations in rodent and non-rodent studies. Also the hippocampal slice assay is used, and that's the current gold standard, as you have defined architecture in the brain. And zebrafish assays are sometimes used too, where people look and monitor the activity of the fish. So here's some advantages and disadvantages of all of these models. I'm not going to go through this in great depth, but a lot of the disadvantages are the same. So concerns around low throughput, animal use, expensive, and then also their translatability to humans. So here I've got some more graphs that I pulled from the paper by Orthier et al. To show how much these methods are actually used in drug development. So on the left-hand side, we can see that rodents are primarily used in seizure liability EEG studies. And then on the right-hand side, the graph shows other in vitro techniques that are used. So I've used the red boxes to highlight all the seizure-related methods. So the hippocampal brain slice assay was used by about a third of respondents, and then stem cell-derived neurons and zebrafish used by about 15% each. However, despite all of these assays that are done, seizure does still persist as a problem within clinical development, which suggests that we do need better methods. And ideally, these will be human-based, which would help us in aiding translation and also reducing animal use. So in terms of identifying seizure risk, are there any lessons that we can learn from the preclinical cardiovascular screening strategy? Now, attrition due to cardiac adverse events has benefited from routine early screening. So specifically, this is cardiac ion channel screening and the use of iPS cardiomyocytes, where you can look at contractility. So the poster child for this is really the hERG potassium ion channel that Helen's already mentioned. So this is a cardiac potassium channel where inhibition is associated with prolongation of the cardiac action potential. This causes QT prolongation in vivo and is linked with the fatal ventricular arrhythmia torsade de pointe. And now that all of this is very well known, everybody knows to avoid hERG. So it is tested within early drug discovery. So people know if they're going to inhibit it, they might drop the compound or change the design of the compound. And benefits of this is that it's automated, translatable, and predictive, and people can design out their liabilities early. So in addition to hERG, there are another 6 ion channels that make up a panel of 7 cardiac ion channels used in screening, called the Comprehensive In Vitro Proarrhythmia Assay, or CIPA. So all of these 7 ion channels are important at various stages of the cardiac action potential, which is shown here on the slide. So for example, influx of sodium is important in phase 0, and that's where we have NaV 1.5, and calcium is important within phase 2. And for that, we have ion channel CAV 1.2, and you can see HERG there marked in phase 3. And then there's other potassium channels dotted around as well. So people look for inhibition of all these ion channels to get an idea of what their compound is doing. This approach has also been modeled and confirmed in iPSC cardiomyocytes. So here I've got some data for quinidine, which is a HERG blocker. So you can see here increases in the field potential duration, or FPD, which links to the action potential duration, and also decreases in spike amplitude, which relate to contractility of the cells. So this brings me on to my important question, which is, could we reduce attrition due to seizure using a similar approach that was used for cardiovascular screening? So basically, can we panelize seizure into a series of ion channels that can be deployed early in drug discovery, and then model this using iPS neuronal cells looking at their electrical activity? So first of all, to do this, we need to look at what proteins are involved in seizure and have a look at the genetic and pharmacological evidence. So the answer to that is that there's a lot of different things involved in seizure. And on the left-hand side of the slide here, I've got a table that I've taken from a paper by Easter et al., where they've looked at the pharmacological targets with a positive association to seizure. So you can see there's many different GPCRs, ligand-gated ion channels, voltage-gated ion channels, transporters, and enzymes. So I've focused in there on the ligand-gated ion channels and the voltage-gated ion channels. Because there's the most evidence associated with seizure, and secondary, we are an ion channel company, so that is our area of interest. And the table on the right-hand side of the slide shows various neurological channelopathies where both gain and loss of function mutations are causative of epilepsy. So a full list of ion channels involved in seizure equates to over 100 targets, which are obviously far too many for any screening strategy. But from looking at the weight of evidence from these genetic studies and pharmacological studies, we've come up with an initial panel of ion channels that may be predictive of seizure as a starting point for testing. Obviously, because we're at the start of this process, once we get more information, we might add some channels, remove some channels, kind of whatever works best, really. So this is our initial ion channel panel. So we've got 3 sodium channels. So these are the 3 channels all associated with epilepsy. 8 different potassium channels. The potassium channel family is absolutely massive. So this might look like a long list, but it's actually quite short. And where there were more channels in one family associated with epilepsy, we just chose one in order to keep the list as short as possible. And then we've got a couple of ligand-gated ion channels as well. I would mention here that SIPA is 7 ion channels, whereas we've got 14 here, so it is quite a lot bigger, but as we're at the start of trying to figure out which ion channels are important, and once we get more information, we'll try and narrow this down as much as possible so people would want to use it. So in terms of validating our approach, we need to pick some drugs. So this is the list of compounds that we selected. It's a fairly standard compound set. Strychnine in there, pucotoxin, different GABA antagonists, etc. Other groups have used a very similar set. For example, the paper by Easter et al. It's a pretty seminal paper looking at hippocampal slice, and also the Hesse New Tox Consortium use a bunch of these compounds as well. So before going into my data, I wanted to briefly mention how we would create the seizure panel and how the screening process works. So first of all, I had to create the cell lines. So we did this by buying in the human ion channel gene of interest and then inserting it into the cells, which are either CHO cells or HEC cells using transfection. And then once we've done this, go through a bit of a long process to weed out all the non-expressors. So we've got some nice clones. So in doing this, we generated 14 separate clones expressing each of our ion channels. We then expose our cells to serial dilutions of our seizure-related compounds, and then look at their activity on our automated patch clamp platforms. So in-house, we have the Sapphire Q patch or the Nanion patch liner, and we use whichever platform kind of made the most sense for the ion channel that I was looking at. So in doing this, we're able to determine compound activity. So here, this is a nice big potassium current. Before drug addition is in the brown, and then you can see when I've added a drug, cause some strong inhibition of the current. And because we do serial dilutions, we're able to get nice graphs like this and then generate IC50s. So here's some example screening data for one of my ion channels, KV 2.1. So you can see inhibition with bupropion, all the compounds down the middle row, and also with Seroquel. And then so I did the screening for all of our ion channels with all the different compounds, which allowed me to make this nice table of summary data. So you can see all the ion channels down the left and the activity with all of our different compounds. So I've tried to do like a traffic light system to help with grading. So anything red is IC50 under 30 micromolar, high risk. Yellow is an IC50 between 30 and 100 micromolar, and intermediate risk. And then anything over 100 we put as low risk, as the IC50 was over 100 micromolar. And we took this grading system from the experience with HERG. So there's a lot of different trends here that I'll quickly go through. So in general, the CNS active therapies inhibited the most ion channels outside of their mechanism of action, illustrating their promiscuity, which was not really a massive surprise. You can see here, that's all the compounds there on the left-hand side. It's filled with red and yellow. But bupropion and quetiapine hit only the KV channels and the nicotinic channels and didn't hit any sodium channels, possibly due to their mechanism of action or structure or something like that. The nicotinic channel was sensitive to the most compounds, and the voltage-gated potassium channels were sensitive to more compounds than the sodium channels. Again, we weren't necessarily surprised by that. And we did see a similar profile with the sodium channels there. You can see on the top there, the 3 sodium channels are hit more or less by the same compounds, which suggests that maybe we could just include one of those and that would be fine. Based on this preliminary activity, it'd be nice to do like a seizure panel light, narrow it down. And I thought this could include the nicotinic channel as it had the most hits, GABA as it's a very important role within the brain, and then a few of the KV channels and 1 NAV. But before deciding that, I probably need a little bit more information to make that choice. The GABA receptor antagonist, picotoxin and PTZ, were very specific to their target, so they only hit the GABA ion channel, and pilocarpine as well showed very strong specificity. Amoxicillin and anoxacin are 2 antibiotics that are reported to inhibit GABA, but they didn't do that in our assay. And something that we're looking at doing at Next is looking at some structure-activity relationships from the structures and potency of all our different compounds of the ion channels, try and figure out if there's any key areas there that are driving this inhibition, which would be really obviously good for people to avoid during drug development and quite a useful thing to know. So what are the benefits of this process? That our assays are automated, functional electrophysiology data, it's high throughput and cost effective, and since mutations can be both gain and loss of function, you need to be able to look at both increases and decreases, which we can do. And as I mentioned, hopefully we'll be able to establish SAR and design out liabilities early, as was the case with HERG. So I'll move on now to our second assay, which is our microelectrode array assay for seizure detection. So here we use IPS neurons that we buy from Fujifilm Cellular Dynamics. And we mix 2 types of cells together, gluteneurons and astrocytes. And the gluteneurons contain both excitatory glutamatergic neurons and inhibitory GABAergic neurons. And then we have the astrocytes in there as they're just nice support cells and just help you get a nice culture. And then we plate these cells at defined numbers and ratios directly on the electrodes, which allows us to look at the electrical activity in the cells. So what you're looking at here is the maturation of the cells over 3 weeks. That's how long I would culture them for before adding any compounds. And each plot here, they're called raster plots, shows the activity on a particular day in one well over 150 seconds. So as I mentioned, there's 16 electrodes in every well, and here each line corresponds to recordings on that electrode. So any little black line is an individual action potential or fire, and then any little blue block is a burst of action potentials. And when all these bursts occur together, that shows that the cells have formed their synaptic connections between each other, and then they begin to fire synchronously, which is what we want to see. So that's happening nicely by day 16 and day 23. We see really nice activity across the well. So I'll show you this in the more pretty format. So here you're looking at day 5, where it's very random activity. So this shows the activity across all the wells in the 24-well plate. Which is nice. And then by day 23, when all the cells have formed synaptic connections, they begin to fire all together across the well. So when you see that big flash, that's when we get those big spikes on the plots, which is quite nice and pretty. So I already mentioned raster plots, but I wanted to just mention it again as it's quite important. It's our main data output. So it shows the activity of the network in one well. We've got individual action potentials, bursts, and network bursts. And what we look for is changes in these patterns when we've added all of our compounds. So here I've just got some example plots of some of our drugs, so some of the CNS active drugs. And it's quite clear to see when there's been a change. So with amoxicillin and paroxetine, you can see this sustained pattern with increased network bursting. So the left is baseline, on the right is after drug. And then with clozapine and bupropion, we also see some changes as well, but more banded. Here we have some of our GABA antagonists. From my experience, bucotoxin is the nicest GABA antagonist to use in our assay. PTZ is commonly used in kindling in vivo, and it's very successful there at inducing epilepsy, whereas in this cell-based model, it's not quite as nice. We do see some changes, but doesn't seem to translate well, not in my hands anyway. And then for amoxicillin and enoxacin, that apparently inhibit GABA, they didn't do anything in the ion channel assay, and they also didn't have any changes in our MEA assay either. A couple other compounds here. 4AP is quite an interesting one. It's a pan-potassium channel blocker and has this nice pattern when we add the compound that we see one big network burst and then lots of small ones, which is quite unique. Pilocarpine was the only compound to cause a decrease in activity, and then also some data here for strychnine and linaperdine. So once we've done all of our drug additions and everything, all of our different recordings, we can pull the data and then look at some changes in different parameters. So there's about 50 different parameters you can look at, which is far too many. So I've just pulled out a few ones here just for the purposes of the slides. So the mean firing rate, the duration of the bursts, the number of network bursts, and the length and the number of spikes within the network bursts as well. So what we found was that the majority of seizure-genic compounds increase the number of network bursts, and that these are shorter and contain fewer spikes. So that's basically on the first part of the table from amoxicillin to quetiapine, and also strychnine has the same pattern as well. Some exceptions include 4AP and pilocarpine. For our GABA antagonists, picotoxin was the sort of nicest GABA antagonist. Amoxicillin and enoxacin showed no effects, and I included acetaminophen as a negative control. As part of doing this work, I did do some comparisons to the hippocampal slice model. Didn't do anything in-house myself, but just looking in the literature to see how it looks compared to our assay, and we can see pretty good corroboration between the two. There are a couple examples, pilocarpine and bupropion. We did see an effect, whereas in hippocampal slice, our effects haven't been observed, but with enoxacin, we didn't see anything in our assay, but something is detected within hippocampal slice and primary cells and other rodent models. So maybe there's a species difference. And I have recently submitted a paper with all of these sort of comparisons in, so if you're interested, please have a look. Just got a slide showing some considerations for setting up these types of assays. You want to be pretty careful with your cell populations. So all the data that I've shown on this slide set contains cells with 80% glutamatergic cells, 20% GABAergic neurons plus astrocytes. And we found that studies with fewer GABAergic neurons, we get different responses. So for example, with pilocarpine, nothing happens. With 4AP, we get a flipped response. And with the GABA antagonists, you really don't see any effects, presumably because of the lack of GABA within the cultures. However, we see more well-to-well variability when you use more inhibitory neurons. So I've just put a couple plots on the side there showing examples of cells with more GABAergic neurons and less GABAergic neurons. So when you have less GABAergic neurons, your cultures synchronize really, really nicely. You get really strong activity. It looks a bit neater, but you don't get you can't see all of your pharmacology. So it looks pretty, but it's not as useful. Other things to consider, as is the case with cardiomyocytes, is differences with batches of cells and differences between suppliers. Also, the timing of dosing is important. You want your cells to be synchronized. And another thing that I want to look at is ion channel expression. With some of the compounds that I've added, the functionality demonstrates expression, but something that we're keen to do is link this together with our ion channel panel and have them as a complementary set of assays. So it'd be nice to see everything that's in these cells. And then before finishing off, I wanted to quickly talk about some work that I've been doing with one of our placement students, where we've been doing some further characterization of the GABA receptor within the neuronal co-culture. So GABA is a pretty big ion channel. It's got a lot of modulatory sites, so it would be nice to look at all of those. So first of all, she looked at the GABA site, adding a commonly used antagonist bicuculline, which did increase activity, which is what we'd expect. It's not that clear from the plots, but when you look at the analysis, you do see increases in different parameters. Likewise, she added some agonists, which we would expect to decrease activity, which they did. She then wanted to see if she could reverse the decreases using bicuculline. She was able to do this with GABA, but not with muscimol, which is quite interesting. Perhaps it's to do binding affinity, something like that. But it was reversible with picotoxin straight away. Another site we looked at was the benzodiazepine site. So here we've used Indoplon, which is a marketing sleeping aid. So you can see here a strong decrease in activity, which was immediately reversed with flumazenil, which is a clinical antidote used for benzodiazepine overdose, so which is pretty cool. And flumazenil alone didn't do anything, which is kind of what we expected to see. So going back to seizure, where do our assays fit into the current cascade? So down the middle of the slide here, you can see the cascade that's currently used, starting off with in vitro binding, followed by zebrafish and brain slice, and then going on to rodent behavior. So what we're hoping is that our assays could go in place of zebrafish and brain slice, therefore reducing the number of zebrafish and brain slice that are used, and hopefully then down the chain, maybe reducing the amount of rodents that are used within this process as well. In terms of how our assays could be useful, we think they could be useful at different stages or used in different ways in different stages of drug discovery and development. So for example, in early discovery within lead generation and optimization, the MEA study might be useful to prioritize drug candidates, and the ion channel panel could be used for early hazard identification and compound redesign. Whereas further down the line, if you're already doing a GLP toxicology and you've seen some convulsions in animals, you could use an MEA study to determine if convulsions are seizure-related in a human model. So do they actually cause changes in brain activity? Because a convulsion could just be due to something else. And with the ion channel panel, you could get some mechanistic insight into the cause of seizures, which may be useful with drug design. So here I've just made up some compounds to show how this could look in practice, compound X and compound Y on the MEA, looking at serial dilutions of these drugs. So early in drug development, you'd probably go with compound X over compound Y because there's less arrows on there. Whereas later in drug development, if you'd seen a convulsion in your animals, you could be pretty happy with compound X, that it's not really a problem when you look at the Cmax, whereas with compound Y, you might be a little bit more concerned. Same thing here for the ion channel panel. I've got compound A and compound B. You would always go with compound B over compound A. But if you wanted to look at mechanistic insight to the causes of seizure, you might know, like, you could find out with compound A, okay, it might be to do with KV 1.1, and maybe there's a way to redesign the compound to get rid of that liability. So to sum up, panel strategy and IPS cardiomyocytes are used for cardiovascular safety screening, has been very useful. In the area of seizure, there's a need and the rationale and technology to do it. So we've developed the ion channel panel and IPS neuronal MEA assay, and the understanding of ion channel involvement is important. And with that, I'd like to thank you for your attention, and I'll leave you with my acknowledgement slide and take any questions. Thanks.
up to the microphones.
Hi, Marian Fernandez, in vitro pharmacologist. Just regarding one of the tables that you show, where there's a lot of variability between the compounds showing differences in burst or frequency, do you think you are in a position to really distinguish what compounds will be pro-seizogenic or anti-seizogenic?
I mean, kind of how we do it is we'll look at novel compounds and sort of compare them to the validation set we've already used. So we can kind of say, oh, it looks a little bit like this, and it looks a little bit like this compound. And we have used a couple negative controls, so we do know what no activity looks like as well. It is a bit difficult because we're still in the early stages, but I think we're confident that we can see something that does cause a pro-seizogenic change.
Thank you.
It looks great, looks very promising. I wonder if you might use these techniques, this panel, for studying anti-epileptic drugs rather than just seizure-inducing drugs. And I'm thinking particularly, for example, of cannabinoids, THC, in epilepsy.
Yeah, it's something that we want to do, so to look at the MEA model and then use it for anti-epileptics. So I'm going to sort of run a study to induce seizure within the culture and then add a known anti-epileptics and see if we can sort of reduce the seizure changes. And it would, be very useful within THC. Something we're looking at the moment. Thank you.
Okay, thank you, Kim.
Thanks.
I'd like to introduce our next speaker. So this is the part of the session where we have oral communication chosen by the programme committee. So one of the highly valued posters, which is also a poster out. No, it's not a poster. It's just going to be an oral communication. So I'd like to introduce Sam Medwid. So she is a research associate at the University of Western Ontario in Canada. Her research interests are pharmacogenomics, and she's going to give us a talk around carriers and polymorphisms, greater risk of fluoropyrimidine associated toxicities.
Thank you. Sorry, it's a very lengthy title, but thank you for letting me speak today. So as it was mentioned, I'm a research associate from London, Ontario, Canada, and I'm going to talk about microRNA miR27A and its polymorphism and the risk of a toxicity in fluoropyrimidine-treated patients. So just so I have no conflicts of interest to disclose. So fluoropyrimidines are a chemotherapy medication. They're commonly used to treat solid tumors, especially in the GI tract. In Canada, we have two types of fluoropyrimidines, 5-fluorouracil or 5-FU, and this is given intravenously, as well as an oral prodrug, capecitabine. So approximately 30% of people that receive these chemotherapies do suffer severe grade 3 or above toxicities. And these include things like diarrhea, neutropenia, hand-foot syndrome, so quite serious complications. Additionally, it can be lethal in certain circumstances. So we do know there is a rate-limiting enzyme in the metabolism of fluoropyrimidines. So this is DPD is the protein name, or DPYD is the gene name. So this is responsible for about 80% of the metabolism of 5-FU into the inactive metabolite 5-DHFU in the liver. In patients, the DPD enzymatic activity is highly variable between patients. However, we do know in patients that are deficient in DPD that they have an increased risk of fluoropyrimidine toxicity. So with that specifically, we know there are certain DPYD single nucleotide variants or polymorphisms, I'll refer to them as SNPs going forward, that are seen in this gene that are associated with toxicity. So numerous groups have looked at this over the past 10 years. So you can see there's two SNPs, STAR2A and STAR13, and they result in no complete loss of DPD activity. However, they are quite rare within the population, less than 0.1% of people. but they do result in a 2 to 4 times increased risk of toxicity in patients that are carrying these variants. Additionally, there's 2 other variants, 2846 and HAPI3, and these reduce DPD activity by about 50%. And HAPI3 is actually quite more common than these other ones, with 2.4% of patients having it. And again, they increase your risk of toxicity. So currently, there are international guidelines available by CPIC and DPWG that suggest that we should preemptively be genotyping patients for these variants before we give chemotherapy medication. That way we can give these patients an appropriate dose reduction in chemotherapy medication that reduces the risk of toxicity but still makes the drugs and the chemotherapy medication as effective in treating their cancer. So with this is a study out of our lab that we've done a few years ago where we dose reduced patients that had these variants. And as you can see now, the relative risk in these patients is completely comparable to wild-type patients. And other studies that we haven't done, but other groups do show that this doesn't increase the risk of cancer recurrence or anything like that. So by giving an appropriate dose reduction, we can prevent quite a few of these toxicities. However, this is limited sensitivity. So we are only able to prevent about 30 to 40% of the toxicities in these patients by doing this genotyping. So one of the things our lab focuses on is what additional causes of variation in DPD activity could be responsible for the rest of these toxicities, and is there a way to prevent this toxicity in these other patients? So one such idea we've thought of is microRNAs. So as many of you probably know, microRNAs are short, single-stranded, non-coding RNAs. They form hairpin-like precursors, which are processed into single-stranded, mature microRNAs, and they generally do bind to the 3' UTR regions of messenger RNAs. And this suppresses the expression of messenger RNAs or blocks the translation of them into proteins. So one such microRNA that has been shown to be associated with DPYD is miR27A. So there is a potential binding site of miR27A on the 3' UTR of DPYD. And some in vitro evidence that has been done has shown that when you overexpress miR27A, it results in the accumulation of risk proteins on the DPYD messenger RNA, and this decreases the level of DPYD messenger RNA as well as protein. Additionally, in vivo, mouse studies have shown that there is a correlation between miR27A levels and DPYD messenger RNA protein and activity. So interestingly enough, though, this microRNA also does have its own single nucleotide variant, RS895819, which I'll just refer to the SNP in miR27A going forward. So this is a variant that's an A to G switch. It has a minor allele frequency of 35%, so very common actually, and it has been shown to alter the expression of miR27A in vitro and in vivo. So one such study by Offer and al. Did show that carriers of this SNP had a decreased DPD enzymatic activity in their peripheral blood nucleocyte cells. So obviously the next question is, does that affect toxicity in patients taking fluoropyrimidines? And 2 studies back in 2015 and 16 did look at this. And they wanted to actually look between DPYD wild-type patients and DPYD variant carrier patients. So this is before those international guidelines started coming out. So these patients were all provided standard dose of fluoropyrimidines. And you can see here in DPYD wild-type patients, if they had this microRNA risk allele, the actual first paper showed that they were at a decreased risk of toxicity, while the second paper showed there is no increased risk. However, what these papers found was quite interesting, is if these patients had a DPYD variant allele, so one of the 4 alleles I had mentioned earlier, they are at a significant increased risk of toxicity by about 4 to 8-fold higher than patients without this microRNA risk allele. So this leads into our hypothesis, is that this miR27A SNP was associated with toxicity in wild-type patients for DPYD variants, but was also seen in patients who had a DPYD SNP and received a genotype-guided dose reduction. So we first wanted to confirm kind of the in vitro findings that I had shown you earlier. So we used an HepG2 cell line model where we transfected in a miR27A mimic for 48 hours. This caused a very high overexpression of miR27A, and then we measured DPYD messenger RNA. And indeed, we saw about a 50% reduction in DPYD messenger RNA, kind of indicating to us that this miR27A is involved in the regulation of DPYD. So to look at this in clinical patients, we asked the question, does this variant increase the risk of toxicity in patients taking fluoropyrimidines, and is this effect seen in DPYD variant carriers after receiving a genotype-guided dose reduction? Because as we know now, many patients are given a genotype-guided dose reduction, so does that prevent the toxicity that those previous studies had looked at? So we used our retrospective cohort that I had mentioned earlier that had been dose reduced. So this was originally 1,300 patients. We only had 45 variant carriers out of those patients, so we used all those and then we matched them 1 to 4 with DPYD wild-type patients. So you can see here the age range, about 62, even number of male and female, quite a few colorectal cancer patients. As I mentioned, fluoropyrimidines are used for GI toxicity, and an even split between the prodrug and the IV drug. In terms of the DPYD genotype, we had quite a few happy 3 patients because that is more common, as well as 2846. Then in terms of their dose intensity of the chemotherapy drug they received, as I said, these are a dose-reduced cohort, so you can see in the DPYD variant carriers, they received about 50% of the ideal dose they were supposed to, as this was an appropriate dose reduction for their genotype. When in the wild-type group, they have about 90% of the dose intensity. And just looking at the prevalence of the miR27A SNP, it's about even between groups, and this is a similar frequency as the literature, about 35%. And now into the actual interesting results. In the total population, in patients that were heterozygous for this SNP, they did have an increased odds ratio of 2.38, so an increased risk of toxicity in patients that had one allele of this microRNA SNP. And then I did stratify my results similar to those other patients. And you can see here in the DPYD wild-type group, nothing was significant, but we did have a p-value of 0.0507, so right on the border of significance for having the variant allele, increasing the risk of toxicity in wild-type patients. But what was really interesting in our DPYD variant carriers that were given that dose reduction, they still had an 8 times higher risk of toxicity if they had this microRNA SNP compared to patients without the microRNA SNP. So that indicated to us that this dose reduction they were receiving was not enough to compensate for also having a microRNA SNP in miR27A. So in conclusion, this SNP may be a clinically relevant predictor of fluoropyrimidine-associated toxicities in both DPYD wild-type and variant carriers. However, this effect was much more pronounced in DPYD variant carriers even after they received a DPYD genotype-guided dose reduction. And our findings suggest that in addition to DPYD, patients may benefit from genotyping for this microRNA SNP prior to fluoropyrimidine dosing to help get a more accurate dose for them to prevent this toxicity. And I would just like to acknowledge the other members of the Personalized Medicine Laboratory in London, Ontario that has helped with this research, as well as our funding partners. Thank you.
Does anyone have any questions for Sam? Can you come up to the microphones, please? Yeah, this one.
Excellent work, interesting presentation.
Thank you.
I'm Rao from University of Geneva. I was just wondering about the last slide where you presented about the odds ratios. The confidence intervals were very wide, and I believe you have adjusted the risks that you obtained with the age of the patients that are included in your cohort, because the p-values and the confidence interval indicates like maybe it's not really the association that needs to be seen.
Yeah, the p-values being kind of close to... Sorry, can you just repeat that again? I didn't...
Maybe if you go to the slide, that's easier for the audience.
This one here?
Yeah.
Yeah.
So if you see the variant allele carriers, so the odds ratio, 95 confidence intervals were very wide from 1 to 86. Could be related to the samples that you, maybe the low numbers that you have, possibly.
Yeah.
Or have you adjusted this association with any other known confounder in the cohort?
Yeah, so we do have low numbers. Obviously, we only had 45 total. And then of that 45, you're looking at a 30% toxicity rate. So it does make that difficult. We did include in here other cofactors. This was a regression analysis with age, sex, as well as DPYD genotype. Because of that difference in DPD activity within genotype, we included that in the analysis as well. However, there may be other factors. We're only looking at 4 DPYD SNPs. These variant carriers are said to have just those 4. They may have other SNPs that we don't know about that are also factors and things like that.
Okay. Thank you very much.
Go ahead.
Hello, I'm François from the University Hospital of Lausanne, Switzerland. Thank you very much for your talk. I wonder whether you might add additional information regarding not only genotyping, but also phenotyping, I mean the metabolites and the primitive substance.
Yeah, that's something we're quite interested in. Unfortunately, this was a retrospective study where we only collected DNA from these patients, so we're not able to do a lot of phenotyping, like that peripheral blood nucleoside activity that I had kind of showed earlier for other groups. Going forward, we're hoping to look more at those things and collecting samples to be able to look at activity of DPD to see if it correlates with the genotype and to see kind of a better in-depth look.
Thank you.
Okay, I think we'll move on. Thank you, Sam.
Thank you.
Excellent presentation. So I'd like to invite our next speaker up to the stage, Maxx Holmes. He's from the University of Oxford, where he's a postdoc working for Blanca Rodriguez in the computational cardiovascular science team. He's working on the development of populations of human biventricular electromechanical models for in silico drug trials. His research interests include development, calibration, and validation of multi-scale models for mechanistic research and unraveling the multi-scale mechanisms underlying cardiovascular disease and toxicity.
Brilliant. Thank you very much, Helen, for that introduction. As you said, my name is Maxx Holmes. I'm a early career postdoc in the University of Oxford in the computational cardiovascular science team. In fact, I've only just been there over a year or so, as of a couple of months ago when I finished my PhD, so very early career. And thank you for SPS for inviting me to give this talk. If you told me 6 months ago I was doing 2 invited talks in a summer, I would have not believed you. So I'm very happy to be here. As Helen said, our team's goal is the development, calibration, validation, and verification of human biventricular models, producing a population of these biventricular models with the goal of running in silico drug trials to aid with potential target identification and improving the efficacy of drug development. So as one of the first sessions, allow me to go through what might be a rather common topic, being the scale of prevalence of heart diseases in the UK. So the British Heart Foundation's most recently published fact sheet in April this year suggested that 7.6 million people in the UK are living with a heart or a circulatory disease. Now, 7.6 million people is quite a staggering number and it's hard to imagine, so I thought what would be an appropriate comparison? And a great comparison would be the city of Glasgow. It's roughly 4.5 times the population of the city of Glasgow living with a cardiovascular disease today. This results in a proportion of all UK mortality across all ages of around 25%. The majority of that is ischemic heart diseases, which I believe since 2008 has been the #1 cause of all age mortality in the UK. And this is equivalent to 180,000 deaths per year, which is more than the population of Oxford every single year. So clearly we need to improve the pharmacological options available. We need to improve patient care. We need to improve the drug development process. It needs to be more efficient. So there's a lot of problems in drug development that we need to tackle, and one of those problems is cardiotoxicity, as has been mentioned previously by Helen. The Pharmaceutical Research and Manufacturers of America have published this handy graphic which kind of illustrates the challenges going from the drug discovery phase through to getting an FDA-approved drug on the market, starting from 5 to 10,000 compounds, narrowing that down to just a couple of hundred in the preclinical stage. And after those 3 to 6 years, we then maybe have 5 go through clinical trials in phase 1 safety, phase 2 efficacy, and phase 3 large population studies. And again, thousands of volunteers, and 6, 7 years later, we may have one drug, which then takes another 1 to 2 years to get FDA approval. And this entire process costs billions of dollars, and that's increasing year on year, with preclinical and clinical both costing obscene quantities of money. And attrition in these drug trials, as has been mentioned, one of the largest causes of that is cardiotoxicity. Cardiotoxicity is the reduction in function of the cardiovascular system, either through reduction of contractility, such as the left ventricular ejection fraction, or the inducements of arrhythmias, such as torsade de pointes. Another reference to Cook, who's done fantastically in this session. It's a major reason behind project closure in both the preclinical and the clinical phase due to safety concerns caused by that. I haven't got the figure, as previous speakers have, but as you saw, it is the tied for first with CNS for the reason why these trials are cancelled, scrapped, or products are taken off the market after release because of these at-risk subpopulations which aren't captured during the initial investigation of these compounds. So because it's quite difficult to investigate all the potential subpopulations which could be at risk during a clinical trial because of the ethical issues and logistical issues of recruiting these volunteers, in silico trials have received a lot of attention for their potential to help with this and supplement existing pipelines to develop drugs in a more efficient manner. So in silico trials is often talked about as the new frontier in digital health. And in silico trials is a bit of a nebulous concept, and one of the main bits of feedback that my group receives when we give these talks is that it's hard to see where in silico trials are going, how are they going to be useful. And so I hope during this talk, can give you a bit more of an idea about that and give you some idea of the progress that's been made in developing in silico trials which are placeable into these pre-existing pipelines. So in silico trials are defined as the development of patient-specific models to form virtual cohorts for testing the safety and or efficacy of new drugs or new medical devices. Is this working? Oh, I'm pointing it to myself. There we go. So what does this actually mean? Well, it means the development, calibration, validation, verification of human anatomy and human physiology. And then only once these models are done can we then start thinking about simulating devices such as a pacemaker, for example, in the context of that human physiology. Or we may want to simulate potential chemical toxicology of a pharmacological compound, again in the context of that physiology. And once we have models for those, we can then move on to simulating treatment outcomes and using large-scale data sets such as the UK Biobank and utilizing machine learning protocols to investigate patient outcomes on a large scale and investigate those in at-risk subpopulations. So how do we actually model a human heart? Well, we start at the smallest scale, the subcellular scale. In 1952, Hodgkin and Huxley published their famous methodology for mathematically modeling the action potential of a giant squid axon. And they did this by modeling the phospholipid bilayer of a cell as an electric circuit. We still use this today, and by using an iterative series of equations, modeling the membranous electric circuit, we're able to produce a cardiac action potential with the major ionic currents all represented, and this is all calibrated using voltage patch clamp data. And then once we have this ionic electrophysiological model of a cell, we can then start introducing the internal calcium dynamics and start relating that to its excitation with excitation-contraction coupling, and finally, active tension, being the mechanical contraction of the heart. But this is just one cell. So when we talk about populations of models, we talk about the same equations used, but variation in these ionic conductances. Variations in the magnitudes of the major ionic currents, the magnitudes of calcium flow in and out of the sarcoplasmic reticulum or in and across the sarcolemma, or the receptiveness of actin and troponin to calcium and how that responds to changes in the L-type calcium current. So where are we now? So We use in each of these stages, all the way from ionic currents, so cellular levels, to single cell, tissue, organ, body. We utilize rich experimental and clinical data sets to simulate human electrophysiology and human mechanics. So I've already talked about the single cell. When we talk about tissue, we talk about rule-based fiber orientation generation. And when we talk about organs, we use imaging data such as MRI to develop these three-dimensional anatomic meshes directly from patients. And when we talk about the torso, the body level, we're looking at ECG recordings for calibration, body surface potentials, and electrode lead placement, so we can then replicate these ECGs in silico. So what we have here is a fully physiologically descriptive electromechanical simulation of the human ventricles enabled by high-performance computing, complete with clinical endpoints, the pressure volume loop, and a fully simulated 12-lead ECG. In addition to this, we can also look at stress-strain relationships and mechanical deformation to investigate their impact on mechanistic arrhythmias. And so essentially we have is a very mature human modeling and simulation technology built from 50 to 60 years of cardiac cell models being developed all the way from the single cell to the three-dimensional organ scale models we have today. But one of the hardest parts about this kind of work is establishing the trust in these models. How do we know that the simulation outputs of these models are representative of the physical reality and not simply a factor or a fragment of the assumptions that go into making these models? You may have heard the old adage, all models are wrong, but some models are useful. And the way we establish this trust with our industrial and clinical friends is by adhering to established paradigms for assessing credibility of computational modeling through verification and validation and uncertainty quantification, also known as VV40, or perhaps using CEPA if we're looking at electrophysiology only, as has been described before. So this is a multi-step process which all starts with defining a context of use for the software we're developing. So in this case, we'd like to run in silico trials in the human biventricular system. So it starts with verification. Verification that the outputs of our simulation match with physical reality. The ECG matches with clinical ECGs. Left ventricular ejection fractions match with the left ventricular ejection fractions of the population, for instance. Or at a single cell level, it could be as simple as action potential prolongation under the impact of dofetilide or renozoline, which is an anti-anginal agent which reduces the late sodium current. Biological relevance of these models. The biological relevance, what we mean by this, is that we custom build the models for the purpose of the context of use. We don't reuse the models and for a different purpose. They are specifically bespoke built to answer a specific question. And they contain all of the physiological information and biological relevancy required to make a good answer for that question. And the next is evidencing predictive capability. I've got an example here, which is done by a fantastic PhD student in our group, Albert Dassey, where he has simulated some clinical trials on our human atrial model, which is electrophysiology only, and demonstrated that for amiodarone, flecainide, and vermicellin, that the simulated outputs match the clinical trials for human across the literature, quite a wide selection of literature, and also that it agrees with animal experimentation in vitro preclinical studies as well. And this is what we need to do for every time we build one of these frameworks. Next is uncertainty, and what we mean by this is doing perturbation analysis, sensitivity analysis, to ensure that we understand how varying the parameters of these models impacts the outcomes. Is it within physical, if we alter, for example, an ionic channel for the ranges of human physiology, will that produce results in the organ scale model representative of human physiology, human reality as well? And at last we circle back to the context of use and we contextualize all of this information for that context of use, and then we have to disseminate that information in a way that makes sense to our industrial and clinical partners. And we've done this previously at the single cell level. This is some work done by Dr. Elisa Passini and Dr. Oliver Britton during their time at Oxford, for which they won 2 of the 3 R's prizes, one in 2014 and one in 2017. And this is the virtual assay software, which is a software package for producing a comprehensive in vitro polymorphism assay in silico. And this was used to demonstrate higher accuracy in using in silico trials at the single cell level than in traditional animal experimentation. So this model produces a population of models by varying those ionic conductances I mentioned before, and applying a compound to that, for example, difetalide in this example, using experimentally reported IC50s and Hill coefficients used from voltage clamp assays. We investigate the drug effects on these ion channels to look at pro-arrhythmic abnormalities and risk prediction. So in 2017, Elisa published a paper which demonstrated a higher accuracy than animal models. We managed to predict torsade the points with a 90% accuracy, which exceeds that of 75 to 85% as reported by Valentina et al. For traditional animal experimentation. This software has been available for quite some years now, and it's undergone a number of upgrades and validation studies throughout the years. And so we've developed that trust at this single cell level. But a single cell level isn't enough to capture the information we need. We need to relate the subcellular mechanisms of the impact of a pharmacological compound with clinical endpoints. So to do this, we start again at the ionic level. We calibrate drug action for a large number of compounds using these reported IC50 values from voltage clamp data, and we apply that to the TOR-RID-LAND human ventricular electromechanics coupled model, which is the new gold standard in human ventricular simulation. From the single cell level, we can look at action potentials, which includes repolarization abnormalities like early after depolarizations caused by difetalide, for example, calcium transients, and active tension. Then we validate this model in both health and disease, and again, under pharmacological action. And then taking into account disease phenotypes, population variability, and utilizing high-performance computing, we translate this to a whole organ model. We do this using the Cristobal Rodero Cardiac Atlas, which was published in 2021, and use this as a basis to then construct the electrical simulation layers and the mechanical fibers layers of the model. And running that under both drug effects and in control, we can then investigate therapy-induced changes in ECG and pressure volume loops. So I'll go through a couple of examples at the single cell level validation that we've done for this, and reproducing a couple of specific drug responses. The first being simple, action potential prolongation performed by renozoline. Again, renozoline is an anti-angina agent, reduces the late sodium current and the Hurg channel. And we can see that in our simulated model, we observe the same dose-dependent increase in early repolarization as is observed in the experimental data for human ventricular trabeculae, which are bits of tissue. Repolarization abnormalities is another large one. Difetalide is famous for causing these EADs in phase 3 of the cardiac action potential, done by blocking the Hurg channel. And again, at the same pacing rates and the same concentrations of drug, we're able to reproduce these EADs in silico, which is fantastic. And lastly, contractility, which is the new one. So for contractility, we're really interested in how the application of these drugs affects sarcomere shortening, affects active tension, and then goes on to affect the mechanics in the whole organ model. So using a number of compounds, including the two calcium channel blockers, quinidine and verapamil, we reproduce these experimental dose response curves for reductions in active tension with concentration of the drug. But there's more that goes into this than simply just a single cell electrophysiological model. There's a lot more. So our team at Oxford has been working on developing a full pipeline for generating these generating these input files to take from a MRI mesh, for example, something you can find in the UK Biobank, running a ECG personalization protocol to match it to a clinical ECG, finding electrode positions and calibrating the mechanical fiber fields of the model, calibrating drug action, and producing all of these fields, for example, transmural heterogeneity, apical basal scaling gradient for the slow delayed rectifier potassium current, fast endocardial activation layers, and transmural varying fiber orientation. Lots to consider. So I won't go into the details of how we validate all of this because that could take several days. But the outcome is that we can then investigate the mechanical performance and the 12 lead ECGs of our digital patient And more importantly, we can output the electrical and mechanical activity maps across the entire heart at every time step of our simulations, which allows us to link those subcellular mechanisms as changes in the ionic currents to the mechanical outcomes and the ECG outcomes that we see in clinical studies. So one thing we also need to take into account is anatomical differences and sex differences in the human ventricles, especially between females and males. So for a couple of case studies for the remainder of this presentation, I'm going to focus on these two hearts. A 59-year-old female heart who weighs 60 kilograms, and a 49-year-old male heart who weighs 90 kilograms. These two hearts were chosen because they lay on opposite sides of the mean spectrum in the Rodera Cardiac Atlas for myocardial volume and for end diastolic volume. So in the middle, we can see that the male heart has a much larger left ventricular volume and a larger right ventricular volume than the female heart, so both cavities significantly larger. And the end diastolic volume, especially in the right ventricle for the male heart, is significantly larger than the female. So we calibrate these models, we run them, and we compare the simulated mechanical biomarkers to the UK Biobank reference populations for men and women of the same age range. So for the female heart, we can see that the end diastolic volume and the end diastolic left ventricular volume is about average. The stroke volume is a bit higher than average, but for the right ventricle, they actually have quite a high end diastolic and stroke volume in reference to the population, which is quite interesting. And then for the male, we see that the right ventricular stroke volume and right ventricular stroke volume is actually rather average, which is contradictory to what we were seeing previously. And due to these factors, the female heart has a much higher ejection fraction than the male in both the left and right ventricle, which, if you looked at just the details from the previous slide, may not make much sense. So it's important that we consider both the anatomical variation and sex differences when we simulate these models. All of this was compared with data obtained by CMR from healthy individuals in the UK Biobank cohort done by Peterson in 2017, which is a fantastic resource that I highly recommend if you're ever interested in population dynamics. It is absolutely fantastic. ECG personalization. So this method was developed by a postdoc in our group, Julia Camps. So what we mean by this is that we match the QRS morphology and T-wave morphologies by inferring the biventricular activation properties from clinical 12-lead ECG signals. So we take a clinical 12-lead ECG, which matches our mesh. Then we produce a population of input variables. These are those fields I was talking about earlier. And we simulate each population to then produce the resulting ECG on that anatomy. Then use machine learning to narrow down the best matches which are physiologically possible and physiologically relevant. And from that, we then have a fully calibrated 12-lead ECG, which matches well with the clinical data. And we check this against every lead, and we also check it against, again, the healthy male and female population ranges for the same age groups. And all of these biomarkers are very consistent across multiple studies. And T-wave progression matches the clinical ECG progression in both the male and female messages, which is fantastic. So a couple of case studies. First, I'd like to talk about verapamil simulations. So verapamil has been described. It's majorly a calcium channel blocker, also minor effects on reducing the hERG channel activity and sodium channel activity. And here we have a nice IC50 curve in dog and human, specifically focusing on the human here. And we can see that on ejection fraction, we see a reduction in ejection fraction as we would expect, dose dependent, with the larger dose reducing the ejection fraction by about 40% of total ejection fraction in females and more in males. The IC50 of verapamil is observed to be about 0.05. Sorry, lost train of thought there. So seeing a reduction of 40% in the ejection fraction with a 0.03 micromolar dosage when we're expecting a 50% reduction at 0.05 fits well with the experimental data. We also compare the PV loops that are generated, and again we can see a reduction in pressure and a slight shifting of volume upwards, which is consistent with hypertrophic cardiomyopathy patient data published by Benau et al. In 1983. The ECG Verapamil very much doesn't have a significant effect on the QRS or T-wave duration, and that's replicated in our results. And we also see a minor decrease in the T-wave amplitude and a minor increase in QT duration in our simulated female heart, and the morphologies match well with the clinical morphologies from Johansen et al. The male heart, I want to bring attention to, and we see a more severe impact in left ventricular ejection fraction at 0.3 micromole than we do in the female heart, which signifies a more severe cardiotoxicity in that heart. There is multiple reasons this could be the case. For example, the much lower starting ejection fraction could make it more susceptible to changes in calcium dynamics. It could also be due to differences in the anatomical data, but this provides an excellent reference point and is exactly why contractility and ECG-based calibration and validation is critical for investigating cardiotoxicity. The next case study I want to take a look at is a little more nuanced. It's a bit more niche. Lead V3 notches in females induced by difetalide and renozoline. So in a randomized clinical study published by Vicente and Strauss in 2015, they presented 2 out of 22 patients, both female, I believe, showed notches forming in the T-wave under the influence of difetalide and renozoline, among other drugs, at their peak concentrations. The example female they presented had quite a small T-wave amplitude, which is quite as the same as in our simulated female. So we're interested, can we reproduce these notches in silico? So we applied the same drug concentrations, matching the pharmacokinetic concentrations, 1 hour, 3 hour, 6 hours post-dose. And we found that in our simulations at the peak concentration, we can then form these notches in the T-wave, which match with experimental data. I think I've run the battery out on that. Which is really interesting that we can reproduce these niche behaviors. We also see QT prolongation under difetalide, and a smaller QT prolongation under renozoline, which again matches well with the clinical data. So that's where we are at the moment. I want to talk a little bit about where we're going next. So this is a simulation of electromechanics in a patient with post-myocardial infarction. So in the previous work with older versions of our model, 2 postdocs in our group, Jenny Wang and Zhen Zhao, produced this study which validated post-MI electromechanics and electrophysiology, the model you're seeing on the left, against acute and chronic stages of post-myocardial infarction. This was on a single anatomy, and we made variations to the border zone conductivities as the condition progresses, and matched them with clinical ECGs and left ventricular ejection fractions. So what we want to take this next is we want to do this, but we want to do it for an entire population. We want to be investigating how variation in scar location, scar sizes, severity of the condition, the stage it's in, and how they interplay with differences in anatomy and electrophysiology and under the investigation of drugs. And to do that, we've secured a lot of new HPC resources through the CompBiomed project, which we're going to be doing this September. And so just to wrap up, talk about the conclusions. So at Oxford, we're enabling in silico trials which can reproduce both the electrocardiographic and the contractile clinical endpoints for organ-scale cardiotoxicity investigations, linking arrhythmogenesis with underlying subcellular mechanisms. The validation is based on both ECG and contractility, and we believe we've shown that it's critical to the study of cardiovascular drug safety and efficacy in silico. And this is something that hasn't really been addressed. You'll have noticed in the first presentation when Helen showed the S7A paradigm, contractility wasn't mentioned. In the C paradigm again, it's not mentioned. It's only recently we've really started taking the idea of contractility-based cardiotoxicity in silico and trying to come up with new methodologies for assessing this. So we've been looking towards medical devices, the VV40 paradigm for validation and verification. The methodologies that we're developing enable investigating sources of population variability, large numbers of compounds with high throughput simulations, and integrations with large-scale data sets such as the UK Biobank. So I just want to thank everyone who's made it possible. You'll have seen plenty of members of my group throughout the presentation, but specifically Blanca, Jenny, Ruben, Doss, and Julia Camps, who made this work possible, and of course all of our funders, and thank you all for listening. I hope you've enjoyed.
Max. That was an excellent presentation. Does anyone have any questions for him? Yes, someone at the back. Can you come up to the microphone, please?
Yeah, it's bright up here.
Yeah.
Hi, it's an excellent presentation. I just want to know, like, what are the inputs that you might have fed to the model before when you were developing the model? The inputs.
So, just to clarify, do you mean the
data inputs which you might have kept? Yeah.
This slide. So, I know it's a bit of a cop-out answer, but everything, really. Of course, we try not to change the ventricular cell model because that's already been well established and validated across multiple studies, and we validated that again with pharmacology, so we tend not to try and change that other than, inducing the population of models through varying the ionic conductances and calcium parameters. The anatomy is what we're investigating at the moment, and we're finding that has quite a considerable impact on the mechanical outputs of our simulations, but also in the difficulty in getting those simulations to work. Not all data is equal, and it often requires a lot of work to adapt a mesh from an MRI from the UK Biobank, transferring it to a point cloud, to then a triangular mesh, to then a tetrahedral mesh, making sure that it's got valvular plugs so we can properly simulate deformation and contractility, and ECG personalization. So the ECG personalization is probably the largest factor, which is why we've spent so much time investing in a method to personalize that on a mesh-by-mesh basis. That alone generates the activation fields you see in the central figure, the transmural heterogeneity, the apical basal scaling gradient, endocardial activation, conduction velocities. That is what I'd say is the largest impact on the end results.
Okay. So this can in future replace the animal studies?
Sorry, can you say that again, please?
In future, can these replace the animal studies?
That's the hope. I don't think it's going to be replacing, but we're aiming to reduce the number of animal studies through these methods. The idea is that in silico trials they can't replace clinical studies, right? We need to be able to do safety studies in humans because we're not going to be able to capture every single detail about human physiology in a model. But the idea is in those preclinical trials, we can run these tests at potential at-risk subpopulations of humans, and that should be able to reduce the number of animals using experimentation. There's also work being done. During my PhD, actually, I did a lot of work on developing animal models to do the exact same work, particularly rabbits I focused on, with the idea of eventually they'd be good enough to replace some animal experimentation. But I think its largest strength would be its contribution at that stage to massively increase throughput and investigate a much larger variety of digital patients than we'd be able to do through in vitro assays.
Last question. Should they be incorporated with the disease animal models also?
So we believe that the human model is the ideal. That is where we want this to go. Like I mentioned, there is some animal models out there. It's not what we work on, but there is some groups who focus on that. Getting the translation from in silico models to the clinic is quite challenging in its own right without including the additional step of going from an animal model to a human, and then to clinical studies. So we believe it's ideal to cut out the middleman essentially in that problem.
Yeah. Thank you. Nice.
Thank you. This side, please.
Yes, I'm François from the University Hospital of Lausanne in Switzerland. Thank you very much, Max, for your brilliant presentation. I have 2 points. How could you integrate pharmacodynamic interaction in your model? And also the second point, the pharmacogenomic aspect of, for instance, HERG and congenital lung QT syndromes.
Okay, so one at a time. So regarding transcription, there's actually a bit of work that's been done recently by Jordi Heimann, who has developed a model for transcription of calcium channel in the cell membrane over the course of a drug's action on the human system. I believe we can couple that model quite efficiently to what we already have, so we can include those pharmacodynamics over time. The one limitation we have in that aspect is that these are obviously quite computationally expensive. We're talking, an entire organ with a high-resolution mesh with millions of cells, and each layer of the simulation adds more complexity. So we wouldn't be able to do it over a time course per se. What we'd be able to do is use the work that's been produced by Jori Hyman to investigate how transcription would affect the cell's ionic dynamics at each time point, and then simulate the heart for a short period at those time points. So, for example, in the work where we're looking at 1 hour, 3 hours, 6 hour post-dose, that's what we'd be able to replicate, those snippets. Could you repeat your second question, please?
Yeah, the second question was on pharmacogenomics, because the higher receptor are associated with a mutation.
Yes.
And there is about 20 different phenotype in terms of congenital long QT, so inherited defect of potassium channel repolarization. So I wonder how you can integrate this aspect in your model.
Of course. So that is kind of exactly where we're wanting to go next. The current work we're doing is funded for Post-MI, but the idea is to be able to expand this to all sorts of genomic preconditions, predispositions to QT prolongation, for example, by mutation of the hERG receptor. But as long as we have the data of how that impacts the conductances or susceptibility, and we understand the relationship between the trigger and the substrate being the anatomy. So, for example, congenital heart diseases, pediatric cardiology, we want to move into these areas to be able to simulate that as well. So to answer your question of how, it would just be a matter of understanding how that condition would affect the susceptibility of the cell to a specific behavior, for example, QT prolongation.
But I mean, you do perform a genetic test systematically on the human heterozygote-related gene or not? This is the question. I mean, you can't just make assumption regarding the pharmacogenomic setting. You have to perform a specific test, genetic test for that.
I think I understand what you're getting. Apologies if I don't. We can have a chat afterwards if you'd like. But that's the whole idea behind the population of models and varying the ionic conductances, is to change the kind of baseline susceptibility to various conditions in the human population to account for factors like that. Obviously, it's not a perfect solution, but for a specific case, we would be able to investigate that.
Right. Okay, thank you.
Thank you. I think we'll move on. Thank you, Max.
Thank you very much.
So I'd like to introduce our final speaker of the session, so Henk van der Linde. He graduated and received his Bachelor of Science from Utrecht Institute in the Netherlands in 1989 and stayed there as a researcher until 1997, where he joined the Preclinical Development and Safety Department at Janssen Research and Development in Belgium. And he's developed to a principal scientist within the Global Safety in Vivo group. So he's a member of the Safety Pharmacology Society and has published many papers around cardiovascular safety models. And so he's really been an in vivo researcher for all of his long career. And so he's going to give us some insight now into new methods and developments to refine and reduce the animal models.
Thank you, Helen, for your nice introduction. Yes, there are my slides. So what we already learned today, I think, because now I make the circle, I think, around, I come back now to Helen for her first presentation. We went over in vitro. That was also what we're using in our department. In silico, we're following that work from Max very closely, and we also do some in silico. And now we're going back to in vivo. What we learned, so the main things are the central nervous system, the cardiovascular system, and the respiratory system. That's what we're looking at. And we should not forget, because that was not here in this presentation yet, that these systems are also connected in the human body. They are related to each other. So that's also maybe why in vivo we have to do some in vivo models. Of course, 3 R's means also replacement by in vitro or in silico, reduction of models. That's maybe can done by multiple measuring multiple endpoints in the same animal models, or maybe even bring these endpoints into toxicology studies. This is the agenda. We will start with Big Brother, so that's something about cameras and the videoing. Then the jackets in your package, that could also be a solution there. Then halfway we have a breath pause. We will look a little bit to respiratory. Then we will put everything together and all together now, so the three main systems. And then the main question, safety farm endpoints in tox studies, is that possible? First, Big Brother. I want just, maybe I should say, I'm not here to bring a show about all the successes we had at Janssen with doing things. Here it's now really what's happening and what are the problems by introducing all these things. So all the things I will tell are studies done by us, models we are trying to set up and we are working on today. So first of all, the home cage analyzer, I will say something about, and also about DSI, do videoing in rabbits. So the home cage analyzer, this is the system at Janssen, and already Helen told you something about what you can measure there automatically, some behaviors. So I don't go to listen them up, but that's what we know. That's also a system we use, but we are trying to get a step further, because we can also, that's what we did, also look at cardiohemodynamic endpoints. So again, put more endpoints in this system by using telemetric, implanted telemetry animals into the home cage analyzer, and to see if we can measure more endpoints in one at the same time in the same animals. So here are some results. So here an A2A-A1 antagonist, we administered it in different doses, low, mid, and high, and the vehicle, of course. And here we see, and it's a Tmax at 1 hour, so it's very left on the graph. You see there an increase in temperature and heart rate. So that's measured by the implanted telemetry. Also the activity taken up by the home cage analyzer, and time spending in rearing and distance. So here we can now measure all these things at the same time in the same animals. So that gives us an idea about the connection between the cardiovascular, the hemodynamics, and with the behavior of the animals. And we reduce the amount of animals we can use. The next thing that was there we have to do also videoing. This is in this case in rabbits, but can also be in rats or other rodents, of course. And here we also bring in EEG measurements. So the animals are implanted with EEG electrodes to have an idea about what's exactly happening in these animals, because you can see all kinds of observations, changes in behavior. Maybe you can see even convulsions or strange behavior, but what is happening on the EEG? That gives us the right indication what we see, we think. So we did this study in white rabbits. I'm not going through all the things, so around 3.5 kilogram, the animals, Charles River, France. So there are, in this case, they were single housed, because we already had an idea what will happen, enrichment and food and water. And here, for instance, this is the videoing of the animals, and at the same time we have the EEG captures. In this case, it was a validation study. Before we went to the real study, we always validate our models. And I just want to show here the PTZ validation. So we made an in-house software that can pick up the EEG analysis. So it can say, okay, the animal is awake. You can see a seizure or a spiking or a sleep wave pattern or all these things. You can go quickly through your signal and then at 2 seconds every time, and the computer will tell you what he thinks or what it is, yeah, and then at that moment you also get the video of the animal. So in this case, this is the normal EEG, nothing strange here. There is some sleep in the animal, and we'll see, but also, for instance, if it's get into a seizure. And I'm not going to show the videos because yeah, that's not necessary. Everybody knows how convulsions looks in animals. That's something we don't want to show you. But here you can automatically quickly go through your EEG signal and directly connect them to the video. The next step, of course, is all three together. That's something we are now at this moment developing, and that will give us a real, a great refinement, of course. So we have the home cage analyzer, so the familiar environment. There is no experimenter is in the house, social housing, but also we have the connection simultaneously between behavior and EEG recordings. You can exactly see on the EEG, is there already a spiking epileptic pre-epileptic event in the animal? You don't see that on the animal by observations, but you already can see it on the EEG. And of course, the animal can be reused for multiple studies. And what also very nice is with the video, of course, you can look back and you can store it and reanalyze it. Normally, we look at the animals and we do an observation. You can write it down, but you never can show the videos again. So that is really an advantage, I think. Jacket in your packet. Here I want to show you two systems we are looking now at. First of all, from 80 cents. That is a system that is developed in rats. It's used already in rats also to measuring ECG, but you can also measure the respiratory and the activity of the animals. So the animals are placed in a home cage. By Bluetooth, the signals are transported to an analyze to the computer, and you could see the signals. So of course, that's free moving, social housed, no surgery, and there is not much stress for the animals eventually when they are used to the jacket, of course, because these kind of things, you need to train the animals before you can use the signals for safety pharmacology. The problem here was ECG is something that is not translatable in rats to humans. So you cannot use ECG in rats for that. You should use ECG when you want to stick with rodents in guinea pigs. So we now are transforming this system for guinea pigs. We are trying that. In this moment, this is still in development. So we are creating now, together, of course, with the company to get new jackets for that to use for the guinea pigs. And this is one study we did. For instance, we did 4 animals with the jacket on. We gave dofetilide and crossover design. We took measurements for 22 hours. The battery is still a problem a little bit. You only can measure for 10, 11 hours, so you have to replace the battery halfway. And here we looked at the parameters, the heart rate, and then the ECG parameters. In this case, it was just to see, are the jackets okay? Can we make changes on it? How are the signals? How are the guinea pigs? They are a little bit different than rats. For instance, they don't have a sleep-wake pattern, but they just sleep whenever they like, during day or night. That's also a main difference between the two rodents. And could we pick up changes? So in this case, we have here in the black dotted line the vehicle treatment of the animals, and in the other colors are the 3 doses of low, mid, and high. And we see here they're in all cases. So we see a decrease in heart rate, for instance, in these animals. And also when we look to the QTC individual. In this case, we used here, that's possible in animals, yeah, that's easier than in humans, so we can do that here. We see in all cases, we see an increase in, we could pick up an increase in QT. Then the next is the jacket. That was in the rodents, and we also do that in non-rodents. So in this case, in dogs that are freely moving, they have, they don't need surgery, and they are stress-free data. This is a known setup. It's used by already more companies. We also use it sometimes. It is something that is, maybe we could bring that to toxicology studies because it's easy and you don't need the jacket system. It will be easy, but it is never a really good looked in relation to the implanted animals, so the implanted telemetric. So what did we do? We had 4 female beagle dogs with around 8 kilograms. We implanted them with DSI software, yeah, to have the gold standard implanted signals, and we put on the jacket and then we both analyzed, brought them together in our Noticard software. That's just a software we use, and we could simultaneously measure that, so beat by beat, to see how looks the signal of the implanted and how is it with the jacket. This case we did moxifloxacin in 3 doses, so low, mid, high, up to 100 milligrams per kilogram. PO, crossover design again, that is the one that we use most to have the highest power in our system, so that is the standard, the crossover, measuring for 24 hours. And the parameters, also here the heart rate, and we look to the ECG parameters and to the activity. I think we only go now to the heart rate. And here we see in red lines, we see the measurements in the implanted telemetry, and the green is the jacket telemetry. And we nicely see over 24 hours. Left upper corner is the vehicle, and then we have the low, and then in the bottom the mid and the high of the compound. You see there are some changes during the 24 hours also in the vehicle. That's normal, but you also nicely see that both systems are measuring exactly the same. So that is really good for the system. Also in this case, I just plotted the QT, for instance. Also here you see that you could measure the QT correctly with the jacket system telemetry as good as the implanted. So the breath pause. Respiratory, we didn't heard a lot about that, I think, today in the safety pharmacology. It is not done as exclusively like the cardiac and the CNS system. So, but there are things going on. So in this case, we looked at the tremor flow. That is in measurements to measure also the lung function. Yeah, there is a little bit misunderstanding when people talk about a volume measuring, about the tidal volume of the lungs, or about the respiration rate. And that is not something that says something about the lung function. That says something about the respiratory function. That is different. In this case, we want to know something about the lung function. And lung function is about the compliance of the lung, for instance, or the resistance of the lungs. So, and that could be possible with this system, because that's a forced oscillation technique. So if we look to the parameters on the right side, you see the respiration and tidal volume and minute volume. That is done by other systems also, but here you can also measure the airway resistance and even other parameters. I'm not going too deep in all these differences, but you can see here there's a kind of loudspeaker generates forced oscillations, and hereby you can measure a kind of flow that can calculate and you can use that to calculate the airway resistance. Okay, that's how it looks like. So forced oscillometry it's called. So you have measurements of 20 breaths, for instance, and then you get the signals. In this case, this is the notochord signals of the pneumotachnograph, because that is standard pneumotachnography in safety pharmacology. And here we combined, or we compared both systems if they give the same result. So we used here 8 dogs. We did in total 120 individual measurements in the dogs. Here I only will talk about the baseline values. For both, of course, we could measure the tidal volume, minute volume, and the respiration rate and compare these. And for the FOT, the forced oscillometry, we were also able to look at the baseline measures for the airway resistance. Again, a crossover design. And here I want to focus a little bit to the power analysis we did. So first of all, if we look to the two systems, we see that the forced oscillometry is always a little bit higher. So in the the tidal volume, the minute volume, and the respiration rate, there is also a little bit increase by the difference in measurements. But okay, that is not a main difference, but we should take account to that. Furthermore, we want to see the predicted sample size, because it's very important in safety pharmacology, or with the 3 R's. We have to see, can we do our measurements correctly at a certain number of animals? Yeah. If you look to the pneumotachnography, for instance, with tidal volume, you have enough for the power with 8 animals, but with the forced oscillometry, you have more than 12 animals needed in one group to do your measurements. Also for the other 2 parameters, you need a lot of animals to do a powered with 80% system. So here the example, for instance, tidal volume, both measured in this crossover study. The tidal volume, we have the different colors for the different amount of persons you can pick up. I know, for instance, for the orange, the above one is to pick up 20% changes on your parameters, yeah, and on the XS, is the number of animals. So you need 8 animals to come with a power more than 80%, something what's needed to have a good results. In the forced oscillometry, yeah, you don't have enough with 8 animals. You will need more than 12. But, of course, the main advantage to use forced oscillometry is that you can measure the airway resistance. And that is not that bad. At least you reach the 80% of power after if you use 12 animals in a group. All together now, yeah, that's a little bit, out of line I'm here, I think, because we are talking about the 3 R's, yeah, but I want to bring it in. Also in this system, here we actually combine all 3 systems, and I think, there are reasons to also to look at this as a 3R model. It is the anesthetized dog mini-pig model. So you would say, okay, that is a terminal model. You analyze, you anesthetize the animals and they don't wake up. But you have big advantages in this kind of models. In this model, for instance, there is no stress or pain at all in these animals. You don't need training. You can give a lot of doses. You can multiple blood sampling without stressing the animals or anything. You can do a wide range of endpoints. Look at the left side. So we're not only looking at QT or QTC, but you can see we do also a lot of work on the hemodynamics, the cardiac outputs, the stroke volumes, but also the contractility, the relaxations, also the pulmonary blood pressures, but also on EEG, on You can take blood samples on the potassium ion in the blood or anything, or on the respiration rate. And here also you can measure nicely the respiration rate or the airway resistance or that kind of things. And you have the combination of the three main systems in the dog. And for sure, we don't use this model a lot anymore, but in some cases we have to do it, I think, on the sake of the 3 R's. For instance, not maybe with echocardiography or speckled tracking echography, we also can do that. In this case, we look to a lot of different risk markers or biomarkers, taking ECSF from the brains, or all these things you can do in these kind of dogs, no problem. But if we think we will get inductions of torsade points, of seizures, we don't want to do that in free moving animals to induce these kind of side effects. So this would be then an ultimate model to use. And then the last question, safety farm endpoints in toxicology studies. I think everybody knows the answer, because the answer is yes, why not? Indeed, we should do that. If we can save animals, if we can save money, if we can save time, we should do that. But there is also a big but, and that's maybe I can explain a little bit more. Then I looked on the computer. I thought, maybe a nice comic drawing would be nice here in my presentation. I couldn't find a lot, but then I crossed this one. It was not the most comic one, I think. I didn't laugh about it, but the first 3 words were very important here. The first day. And that is the main challenging to bring safety farm endpoints in toxicology studies, I think. Because the first day is a very important day in safety pharmacology. We already heard, we looked at the first 24 hours when we induce a single dose of a compound, and there we look at the side effects, not the adverse effects from changing after 3 months high dosing in toxicology, tox doses. No, we're looking at doses 30 times the efficacy dose normally, and there we look at the side effects at the 3 systems. So the first day should be very a quiet day, actually, for safety pharmacology, with not too much interference of humans. But in tox studies, it's a little bit different. That's also the day where we take the blood sampling, doing the body weight measurements, and all these kind of things. So at that time, actually, we need a very quiet room, limited access, nobody can enter. Yeah. To avoid the disturbance and the impact of the measurement parameters. You already saw from Helen, I think it was, with when you take one blood sample, what happens with your blood pressure. That's something you want to avoid. Also, what's very important, you have to train all your animals. For instance, if they have a jacket on, that's something you maybe want, so you have to train all your animals and do acclimatization, and doing also measuring all these baseline measurements of your parameters. For instance, because you have very sensitive, you have to pick up very small changes in certain parameters. For instance, in QT, that's why the question about variability, but also about blood pressure. I come to that back later. Also, we need, because we are going very high in tox studies, we have to be very careful that the toxic effect at a certain moment, every compound has an effect, I think. Even water, if you take too much in it, it's also toxic. That it not interfere with your cardiovascular or respiration endpoints. And then, Then the N numbers, that is also a problem. I already show you if you want to measure proper respiratory endpoints, you need to go to up to NS12, and that is something you don't do in tox studies. There are sometimes NS4 or NS2, 2 males, 2 females already is in the groups are used. And here it's also feeded with the threshold, and that's maybe also an answer on your question a little bit. But of course, QT was important 20 years ago when compounds withdrawn from the market because induction of torsades, and I think we solved a lot there within safety pharmacology. But the hemodynamics now is getting more attention, and we have to pick up actually 3 millimeters of mercury on the blood pressure in our animal models, because 3 millimeters of mercury over 10 years or over 5 years in humans can make heart failure in these humans, and that's why now also it's looked so closely to the hemodynamics of our new compounds. But give you an example here, if you do a dog telemetric study with moxifloxacin, 10 milligrams per kilogram, you can pick up 5.9 milliseconds of your QTc. The FDA wants you to below 10 milliseconds to pick up that, so that is okay. Also in the TQT study, it's possible. So that's maybe an answer on your question. The variability is there also okay. But if you do it in a tox study, a repeated dose tox study, you see no effect of 10 milligrams per kilogram. Even after 30 milligrams per kilogram, you see no effect if you do a normal measurement of your QT. But you need even 100 or 90 or 100 milligrams per kilogram to pick up a certain effect because of the low power of your system at that moment. Okay, and then the last one also we have to take care of the type of instrument we should think of. If you do a mainly restrained, so a snapshot quality measurements, that is not doable for safety pharmacology. The jacket system I showed you, that could be a very good one to use in our tox studies if we train the animals and use correctly. The implanted telemetry, of course, that is the golden standard at this moment, but of course complicated. The dogs after 3 months they are also terminated for histological and everything in these tox pathology studies. So that's also something you not want to do. And the last thing I think here, that's the parallel. The tox studies are mostly in parallel studies, and that is something we want to avoid in our safety studies. We mainly do their crossover studies design. So I think that's the end of my story. So if there are questions.
I don't think there's any questions. I think everyone's looking to go for their lunch.
It is, we are over time or not?
So, so thank you very much again to all our speakers and for the audience that have participated in this session. And thank you.